My name is Robert Bissonnette. In the next 10 minutes, I'll do a brief overview of the key elements to consider when designing clinical trials with systemic drugs in atopic dermatitis. This is my disclosure slide. Let's start by phase 1. Phase 1 first-in-man studies require very close monitoring after first drug administration. These are usually conducted in specialized facilities where patients are confined for a few days after the first drug administration. Should these be conducted in atopic dermatitis patients or healthy subjects? This is a question I often hear. We need to remember that those first-in-man studies can be dangerous. This is a publication from a study conducted a few years ago with a drug that was intended for neuropathic pain. The study was conducted in healthy subjects. Preclinical studies revealed no specific safety signal. We moved on to the first study in humans, healthy subjects, given a single ascending dose of 0.25 to 100 milligrams, no safety issues. The multiple ascending dose phase of the study, where subjects received a daily dose for 10 days of the same dose, revealed no safety issues from 2.5 to 20 milligrams. However, when the 50 milligram dose was administered to the first subject, after 5 days, that subject complained of blurred vision, dysarthria, gait disturbance, had a stroke, was admitted to the hospital, and died 3 days later. It turned out that 4 out of 5 subjects who were treated at that dose had neurological symptoms. So my answer to that question is, I think it's better to do phase 1A in healthy subjects, and when safety information is obtained on the drug, move to atopic dermatitis subjects in phase 1B or 2A. There are some disadvantages of doing phase 1A in atopic dermatitis patients, including increasing costs, delays in completing the study, risk of lower quality efficacy data, because these studies are often conducted in facilities where the PI is not a dermatologist. What about phase 2? Phase 2s can be divided in 2A, proof of concept study, and 2B, dose ranging. My suggestion is for drugs where the drugs that are targeting a new molecular pathway that has not been shown to be a pathway that induces improvement in atopic dermatitis, my suggestion is to do a phase 2A as opposed to a large and costly dose ranging studies. Randomization can be 1-to-1 or 2-to-1, 2-to-1 being better for patients. Now, 2 key questions: how many patients and which disease severity? In terms of how many patients, I've asked our statistics group to do a sample size calculation based on data from 2 different systemic drugs on the market or developed for atopic dermatitis, upadacitinib and dupilumab. I've asked them to use these parameters, specifically a power of 90%, which is a little high for phase 2A, and to calculate the number of patients needed. And this is the result of that calculation. So to detect upadacitinib-like efficacy with an oral drug, one would require about 30 patients with a power of 90. And to detect dupilumab efficacy, it's about 100 patients. So the answer lies in between those numbers. Sometimes I've been asked to do a 10 or 12 patient proof of concept study with atopic dermatitis, randomized 1-to-1. This is definitely too low, but you don't need hundreds of patients. Which disease severity? The 2As are often conducted in patients with BSA 10+, EASI 16+, and IgA 3+. Same patient population as in phase 3. However, they can also be conducted in patients who have less body surface area involved. This very recent interesting publication from Dr. Gutman's lab compared the group of patients with quote-unquote mild atopic dermatitis, defined as BSA of less than 5, who had never had more than 10, to patients with moderate or severe atopic dermatitis, defined as BSA 10+, and differentiated with SCORAD. So biopsies of this trial showed that even in the quote-unquote mild patient, those with a BSA of less than 5, there was significant Th2 and Th22 activation, as well as Th17 and Th1. So there are advantages of conducting trials, phase 2A trials in this patient population. In addition to all the axes being activated, the enrollment is usually faster, requires fewer sites, lower cost, more tier 1 sites. So sites that are known to do very good work in terms of atopic dermatitis studies are usually very busy, and often they will turn down those to a study because they have too many competing trials. Phase 2b, dose ranging. The goal is to study safety again, but to try to find what is the best dose to bring to phase 3. A few special considerations. Open label extension, very important in order to be able to enroll in those trials. Patients are much more inclined to participate if there's an open label extension. Rescue therapy is becoming more and more a hot topic. We've seen that in trials where rescue therapy is allowed very early, that the effect size seems to be lower. So I suggest to have a period of time where patients are not allowed rescue therapy. If they need treatment, then it's an early term, and they go on to be treated with what is needed. IEC has a position paper on this and suggests 4 weeks before allowing rescue therapy. When to include children in systemic clinical trial designs? Well, FDA in 2018 had a guidance document which says as early as possible. So as systemic drugs are riskier than topical drugs, what is usually done is that in phase 3, we see studies with often adolescents, and younger children are usually exposed to the drug once safety and efficacy has been shown in adolescents. So this is often either in a parallel trial that is started a little later in drug development, could be at the same time the phase 3 is done, or it could sometimes be after the drug has been approved in adolescents and adults. Now, phase 3, the design is fairly standard and often imposed by regulatory authorities. They require 2 identical pivotal trials with systemic drugs and atopic dermatitis. Which measures to include? This publication from Home is interesting because it compares various measures that are currently used in atopic dermatitis studies and will help by giving information of what the measure can actually detect. So this can be used in order to select which measures to use. Finally, a few key elements in phase 3. Claims must be substantiated by patient-reported outcome, meeting FDA requirements, so it's not any type of patient-reported outcome that can be used. One very important key element is that a dose will be approved specifically by FDA if it has been tested in the placebo-controlled phase of phase 3 studies. So if the dose is not certain following phase 2, it's often a good idea to study 2 doses in phase 3, which will give the opportunity of filing for approval of 2 doses and will give dermatologists the opportunity to use a lower dose and a higher dose in patients. Thank you.